Tonight, we have invited Professor Itamar Wilner to give the fifth lecture of SMAT talk. Professor Itamar Wilner worked in Israel. He was selected as a foreign member of Chinese Academy of Science last year. His research interests including supramolecular chemistry, nanobiotechnology, molecular self-assembly, molecular and biomolecular machines. Actually, he is one of my supervisors. In 2005, I worked in his group for 3 months. Now, he served as a member of the advisory board of supramolecular materials. SMAT Talk is a senior of online talk organized by the editorial team of the journal Supramolecular Materials. Supramolecular Materials is an open access journal that provides a platform for the innovation research of supramolecular science and material science. The journal publishes high quality original research results related to supramolecular materials, including both fundamental research work on supramolecular science and functional material system. The journal is measured and operated by KE Publishing Group. I believe the journal Supramolecular Materials will have a good future. Now let's welcome Professor Itamar Wilner to give the talk. Please, Itamar.
Thank you very much, Professor Shifang. In fact, I'm very pleased to present this lecture. Unfortunately, I have to do it by Zoom and not as a frontal lecture, but I hope that you will enjoy and you will be able to follow it. Now, my talk will discuss DNA switches and machines, and from the DNA switches and machine to dynamic DNA assemblies. Now, in contrast to chemical reactions that are performed in the test tube and operate based on molecular or supramolecular interactions, the chemical processes in nature are much more complicated and operate on different principles than normal chemical reactions in the test tube. They perform quite complex processes such as cell motility, cell proliferation and differentiation, signal promotion, gene expression, and so on. And all these processes are based on complex networks that intercommunicate one with another. The principles of the native networks include several very basic concepts. First of all, they are triggered by signals and represent stimuli-responsive systems. The networks are dynamically equilibrated networks, and in such cases, they reveal certain properties like adaptive, hierarchical adaptive, feedback-driven, and intercommunication cascaded properties. All these functions will be discussed and highlighted in my presentation. Many of these networks are not equilibrium and are not operating under equilibrium conditions and are out of equilibrium transient network. And also the transient networks are triggered by auxiliary stimuli. They reveal oscillatory or switchable features, gated and cascaded transient operations, and dissipative operations that reveal emergent functions. All these properties will be discussed in my talk. In fact, these topics belong to a rapidly developing area in chemistry that is called systems chemistry. And systems chemistry represents an effort to mimic native dynamic systems and networks by artificial chemical means. During my talk, I will discuss two very basic concepts. One, dynamic DNA networks and their applications. And here I will address and introduce constitutional dynamic network and dissipative out-of-equilibrium network. In the last part of my talk, that will be shorter, we will discuss dynamic machines, DNA machines, and DNA origami structures, and I will try to introduce how we can apply them. Now, what is a network? And this is basically the most important slide of my talk. If we understand the operation of this kind of network, we can then follow all the other slides and all the other systems that I'm going to describe. We see here in the middle a network that consists of four constituents: A prime, B prime, BA prime, and BB prime. These four constituents, each is composed of two components: A prime, BA prime, BB prime, and so on. Now we can see here that, for example, A prime shares components with AB prime and BA prime, but it does not share components with BB prime. The same story is here. You can see that AB prime shares components with AA prime and BB prime, but it does not share components with BA prime. Now, all these four constituents exist in equilibrium. Let's say that we take a trigger T1, and T1 stabilizes from some reason, let's say constituent AA prime. If it stabilizes AA prime, this entire equilibrated system will be reconfigured to a new network where, because of stabilization of AA prime, it will have to increase its constant because of the stabilization. How will it increase its content? By separating AB prime and BA prime to provide the components A and A prime to enrich this constituent. But if this constituent is enriched, and these are basically reduced in their contents, B and B prime are released, and they will form B and B prime as a constituent. So what we can see is that if we stabilize and upregulate one of the constituents because of stabilization, all other constituents that share components with the upregulated constituents will be downregulated, AB prime and BB prime. But concomitantly to the enrichment of AA prime, we enrich the constituent that does not share components with AA prime. If we understand this principle of a constitutional dynamic network, we are ready to understand all what I am going to describe in this area of constitutional dynamic networks. Now let me introduce a simple example, and this example is based on chemical constituents, And it is an example that was introduced several years ago by Jean-Marie Laine. It includes 4 constituents that are displayed here in red, yellow, green, and blue. The constituents include a pyridine unit or a phenyl group as component, and a dimethylaminobenzene component and a phenyl component as components that combine the constituent that is basically an acyl iminohydrazine. All these are acyl iminohydrazine, and they exist in equilibrium. You see here schematically the equilibrium of these components. Let's say that we add as a trigger metal ions, and the metal ion will bind to the red constituent. It forms a complex, it stabilizes this constituent, so the component M will be upregulated. It will increase in its content, but it will increase its content on the expense of the 2 constituents that share components with the red constituents, these 2. This will be downregulated and have a lower content. But concomitantly to this process, the constituent that does not share components with the red one will also be enriched. So this happens when we add the metal ions. But let's say that we, instead of metal ions, we take this network and we shine light on it. This trans-imine bond will isomerize under light condition to the cis state. In the cis state, the component, the green constituent, will be stabilized by hydrogen bonds here. If it is stabilized by hydrogen bonds, the green constituents will be upregulated together with the yellow that does not share components, and the 2 constituents, the red and blue, that share components with the green one, will be downregulated. So this is an example how a network adapts itself to an external signal, metal ions or light or any other. Many such systems were developed, but they suffer from one limitation. Each system, each functions, we have to design a new system, and we don't have a comprehensive material to apply to develop such networks. And here comes our idea to use DNA as a versatile material to develop such networks. Now, how did we come to this idea? We were very active in developing DNA machines. We were very active in developing objects that are modified by DNA and have some motility properties like aggregation, deaggregation. And this led us to the possibility to use the DNA as a functional material to develop dynamic network. What am going to do in the next minutes is to describe some networks that are based on DNA and to discuss with you possible applications of such systems. Now, why do we want to use DNA? DNA, we can encode a lot of information into the base sequence of DNA. Besides base pairing formation of duplexes, we know that we can stimulate structural and functional features into the DNA material based on the base thickness. For example, we can take duplexes and reconfigure them into triplex structures. We can take single strands that are rich in G bases, and in the presence of potassium ions, we can generate G quadruplexes. In the presence of crown eaters, we can separate the G quadruplex to this system. We can take single-stranded nucleic acids, bridge them by metal ions, and separate them by ligands. For example, CC bridges, CC bases, we can bridge by silver ions and separate them by cysteine. TT bases, we can bridge by mercury ions and again by cysteine, separate them. We can take, for example, strands that are stabilized by functionalized with azobenzene, photoisomerizable azobenzene, while cis azobenzene does not interact with the DNA. Photoisomerization to the trans will intercalate the trans isomer and stabilize duplexes. But if we reisomerize to the cis, we can separate the structures. So you can see that we have a whole battery of principles to separate and to form and separate or reconfigure DNA structure. In addition, we can have functions. For example, we can design cofactor-dependent DNA enzymes that reveal catalytic functions, and the DNA enzyme can be a metal ion cofactor-dependent DNA enzyme, or a DNA enzyme formed on a reconfigured structure of DNA, like a G quadruplex that binds hemine and generates a hemine G quadruplex DNA enzyme that has peroxidase activities like oxidation of amplex red to the fluorescent resofurin by hydrogen peroxide or the generation of chemiluminescence. So let's go and describe the simplest constitutional dynamic network that is shown here. It includes four constituents, A prime, B prime, A prime, B prime, and B A prime. The scaffolds of these constituents represent the functional unit that will be reconfigured by the trigger. Each of these constituents is conjugated to a DNA enzyme. And in this case, it is a magnesium-dependent DNA enzyme. This magnesium-dependent DNA enzyme acts as a transducer to tell us the content of the component in the equilibrated mixture. This DNA enzyme cleaves a fluorophore quencher modified substrate. The rate of cleavage is proportional to the content of the DNA enzyme. And if we have an appropriate calibration curve, the response of this transducer unit will tell us how much material we have in the network. So let's say that we have this constitutional dynamic network with this structure. And now we add to this a trigger C prime. The C prime will displace the strand C, and we engineer this constituent in such a way that after displacement, this strand of constituent A prime will form a triplex TAT structure. If it forms the TAT structure, it stabilizes this constituent. If this is stabilized, the constituents that share components, this and this, will reconfigure into lower concentrations. On the other hand, B prime that does not share components with this will be upregulated and will increase in this mixture. Indeed, this is exactly what happens. What you see here are basically the responses of the DNA Zyme units. I'm not going to describe them in detail, but after analyzing them, AA prime increases in the reconfigured network by about 200%. This one increases in content by about 150%. And these two constituents are downregulated by about 70 or 80%. So you can see that the network follows the adaptive features that I introduced at the beginning. One simple example that will, again, identify this adaptivity properties is shown here. We have a network X. These are the four constituents. We have an auxiliary strand C that is modified by innocent cis-azobenzene unit. If, in this case, when we shine light on it, and we convert the cis-azobenzene strand into trans, it will stabilize, in this case, a duplex with this part of strand BA prime now. It will open the triplex and will stabilize here a duplex. But note that at the moment that this was reconfigured by light to this, we lost the stability of the triplex. So this became now, the constituent BB prime is now weaker, less stable. It is downregulated. The AB prime is also downregulated. But those components that share the constituents that share components with this are now upregulated. If now we take the reconfigured network Y and we shine light on the system at 365 that converts the trans-azobenzene to the cis, we separate this component and we regenerate the network X. So we have by light a reversible reconfiguration of the network, and this is exactly what happens based on this adaptivity principle. So if we realize that we can generate adaptive network, we can now move on to networks of higher complexity that mimic the properties of natural networks. And I will move on to show you that we can generate intercommunicated networks. For example, if we have two networks, S and T, and we introduce into this mixture of two networks a hairpin structure HAA prime that is being cleaved by this DNA Zyme. We engineer this hairpin in such a way that the cleaved fragment HAA-1 stabilizes this B-loop region of the constituent AA prime in network S. You will immediately say, if we stabilize the triplex structure here, this will be upregulated, this will be upregulated, and these two will be downregulated. But in networks T, nothing is expected to happen. This is exactly what you see. AA prime and BB prime are upregulated by about 140 and 90%. The other two constituents are downregulated, but the network on the right side is not affected. We can move on and take this mixture of two networks, and now we subject the two networks to hairpin HDD prime that is being cleaved by this constituent, BB prime. We generate a fragmented product that now stabilizes this B loop region. If it stabilizes this bi-loop region, it will upregulate, increase the content of this constituent, and these two will be downregulated. Nothing will happen on the constituents of the left side. This is exactly what happens. You see that DD prime and CC prime are upregulated, while the other two are downregulated. So if we understand that, we can now take the two hairpins and subject the two hairpins on the two networks, and now we have intercommunication between the two networks, and you see this network feeds all the time network S with this fragment HAA prime and enriches these two components, and network S generates all the time these fragments that enriches CC prime and DD prime. And this is exactly what happened. You see that these two are temporally, they reveal a time-dependent increase, the other two are decreased, and these two are again increased, and there are two other constituents decrease in content. So this is a nice example of intercommunication between DNA-based networks. Besides intercommunication, I mentioned the feature of feedback. Here we can see a nice example of a network based on DNA that reveals feedback properties. We have here a network X. We subject the network X to this hairpin H1. H1 is being cleaved by this DNA Zyme units that is associated with the constituent BB prime. We generate this strand. This strand is a subunit of a G quadruplex. Everything is pre-engineered in this network so that this subunit will react with these fragments of AA prime and form a G quadruplex here. But formation of the G quadruplex on this constituent will stabilize this constituent. If this constituent is stabilized, it will be enriched. But if this is enriched, we know already that B beta prime will be concomitantly also enriched. If it is enriched, more of this hairpin is being cleaved. So we have a continuous feedback of the network that will induce a time-dependent feedback-driven enrichment of this, and a time-dependent feedback decrease in the contents of these two constituents. This is exactly what happens. You see here a feedback-driven enrichment of AA prime and BB prime, and a time-dependent decrease of the other two constituents, BA prime and AB prime. So until now, we have seen that we can communicate networks, we can have feedback-driven networks. We were able to show that we can also induce substitution and emergent functionalities, but we thought that it will be good to go one step further and to enhance the complexity of the networks. Until now, all the networks included 4 constituents. It was a 2x2 network. One can think to make networks of enhanced complexity, for example, 3x3. Now, what I'm going to show you is that if we go from a 2x2 to 3x3, we can induce a new functionality into the network and not only demonstrate adaptivity, but we can demonstrate hierarchical adaptivity. What do I mean by hierarchical adaptivity? Here now I show you that we can generate a 3x3 network that has now 9 constituents in it. If I stabilize with T4, a signal T4, for example, the red constituent here, all the constituents in this complex structure that share components with red, this is this, and this, will be downregulated. On the other hand, the constituents that do not share components, these are this and this, will be upregulated. So you can see that you can now generate a new network with 5 constituents that are upregulated. But now if we will apply on it T6, from this we can now upregulate only these 3 and not the other. So by applying on a complex structure several triggers, you can induce hierarchically enrichment of certain constituents in the network. And believe me that these results that are shown here explain or demonstrate exactly these properties. And we were able to show that we can induce hierarchical transitions in these 3x3 networks of enhanced complexity. But you might ask a very legitimate question. You show us here this complex network. For what is it good and what can we do? What application can emerge from this dynamic network? And in the next few slides, I want to show you a few applications. The first application is to control biocatalytic cascades of enzymes. Nature operates in confined cellular environments. This leads to very effective biocatalytic cascades. We have one enzyme that generates a product that acts as substrate for the second enzyme, and if they are at spatial proximity, the cascade will be effective in the confined media. In fact, 2 years ago, we published in Nature Catalysis a review article on confined environments that enhance the cascaded biocatalytic process. But let's say that we have a constitutional dynamic network that is composed of components where one component is modified by glucose oxidase and the second component is modified by horseradish peroxidase. In this case, for example, constituent AA prime will have in spatial proximity these 2 enzymes that, because of the spatial proximity, will induce the biocatalytic cascade. Glucose will be oxidized to gluconic acid and hydrogen peroxide, and hydrogen peroxide will be effectively supplied to the horseradish peroxidase to oxidize ABTS to the colored product that will follow the biocatalytic cascade. This will happen in the constitutional dynamic network M. But what will happen if we trigger this constitutional dynamic network with a strength T1 that will stabilize by triplex formation the DNA scaffold of the network? Under these conditions, this will be upregulated. And if it is upregulated, we enrich the content of the two enzymes in the spatial proximity. So we will enhance the biocatalytic cascade. This is exactly what happens, that we make an improved biocatalytic cascade. If we reduce the content of this constituent, we will downregulate the biocatalytic cascade. And this is exactly what was demonstrated in this paper that was published about a year and a half ago in Nature Catalysis. Now, this is a very general phenomenon, and we were able to show that we can control and regulate such biocatalytic cascades for enzymes with a cofactor, for example, alcohol dehydrogenase and NAD. Upregulation by the network improved this biocatalytic cascade, whereas downregulation of this constituent led to a lower activity of the system. We can move on and show you that we can use this control of chemical processes by the network by designing an artificial photosynthetic system. And again, what we did here, we had a network where one of the constituents, again, this A prime, is functionalized with a zinc protoporphyrin G quadruplex photosensitizer, and the other component with a B-pyridinium electron acceptor, the V2+. In the configuration at equilibrium in the state X, the photoexcitation of the zinc chromophore leads to electron transfer to the bipyridinium salt. We make bipyridinium radical cation that in the presence of ferredoxin reductase takes NADP and makes, as in normal native photosynthesis, the NADPH. But here we have a certain activity of the photosynthetic, artificial photosynthetic apparatus. What will happen if we apply a trigger T1 that enriches AA prime? It makes stronger this AA prime, but the trigger forms a triplex structure with this AA prime. And because of the triplex structure, AA prime is enriched. Under this condition, the photosynthetic, artificial photosynthetic system will be enhanced because of the enrichment of the constituent AA prime. If we downregulate AA prime, certainly the photosynthetic system will be downregulated. Here you can see very nicely that you can control indeed the photosynthetic system by the DNA scaffold that dictates how much of the active photosynthetic apparatus we have in the system. Let me move and show you how we can use the DNA-based network in order to control drug release from a carrier. Now here what I'm going to show you is not only that we can dictate what drug we release, but also the quantity of the drug that we release from the system. What we did is we generated or synthesized two metal organic framework nanoparticles. We loaded one metal organic framework nanoparticle with doxorubicin, an anti-cancer drug, and the second metal organic framework nanoparticle with the camptothecin chemotherapeutic drug. The entrapped drug were locked in the MOF structures by duplex DNA structure. This is a methodology that was developed by our laboratory to lock and unlock drugs in nanocarriers. Now let's say that we take our network and we take the network under these conditions. Under these conditions, if we subject the network to two biomarkers, microRNA 124 or microRNA 125, under these conditions, if we add to the network, to the parent network, two hairpins, H1 and H2, the constituent AB prime and the constituent AA prime will cleave these two hairpins, generate fragments that will unlock the nanomorph and release the drugs. So under these conditions of the parent constitutional dynamic network, we have no selectivity. The two drugs are released in an inefficient way that is demonstrated by the black curves in the two release pattern curves. If we subject the network, the parent network, P, to microRNA 124, that is a marker of a certain kind of cancer cells. It will stabilize the constituent AB prime. This will increase the content of this constituent, will enhance the cleavage of this hairpin, and the released fragmented product will unlock this MOF particle and induce the release of camptothecin. You can see here the release of camptothecin. Under these conditions, the release of doxorubicin is very inefficient because the content of AA prime is here very low. But if we take this constitutional dynamic network and we subject it to microRNA 155, we stabilize now AA prime. AA prime will effectively cleave BB prime, unlock the DOX-loaded MOF nanoparticle, and will release effectively the doxorubicin. We see here that under these conditions of this microRNA, we have effective release of doxorubicin and very inefficient release of the camptothecin. So what we learn here is that the constitutional dynamic network can regulate the release of drugs from drug carrier, and it acts as a dynamic controller of the release process. Let me move on and show you that we can control functions of materials by this constitutional dynamic network. And here what we did, we controlled the stiffness of a hydrogel by the constitutional dynamic network. We have a mixture of 4 constituents, AA prime, BB prime, BA prime, and AB prime. AA prime and BB prime are Y-shaped DNA constituents. In contrast, the constituents BA prime and AB prime are bifunctional Y-shaped constituents. All of the constituents include toehold domains that provide complementary sequences that can be bridged between the constituents. So if we mix this equilibrated mixture of constituents, we form a cross-link network of these DNA subunits, and this forms a hydrogen. Now note that the simple Y-shaped constituents, they don't form cross-linking units. Only the bifunctional Y-shaped components form cross-linking units. So the ratio between the Y-shaped and the bifunctional cross-linking Y-shaped units will control the stiffness of the gel. More of the bifunctional bridging units will introduce higher cross-linking and a stiffer hydrogel. Lower bifunctional cross-linking units will generate a hydrogel of lower stiffness. How can we control the stiffness? If we add a strand, for example, E prime, and this E prime stabilizes the biloop region of the double bridged units. We stabilize these units, we will increase in the equilibrium the cross-linking components, and we will get a higher cross-linked hydrogen. In turn, if we take this hydrogel instead X, and we stabilize the constituent of the simple single Y-shaped units, we will get lower cross-linking and a hydrogel of lower stiffness. So we can have here hydrogel at 3 degrees of stiffness. X is anticipated to show the medium stiffness, Y higher stiffness, and Z lower stiffness. And the stiffness is controlled by these triggers, E1 or E2. E1 should make a higher stiffness, and E2 a lower stiffness. And if we apply counter triggers, we certainly can switch the reversible stiffness properties of these hydrogen metals. This is what happens exactly in the experiment. You can see that microindentation studies demonstrate that the hydrogel in state X that has the medium stiffness shows about a Young modulus of about 13,000 Pascal. On the other hand, when we apply the strand E1 that makes higher stiffness, we almost double the Y modulus of the hydrogel, demonstrating its higher stiffness. If we apply the strand E2 that stabilizes the simple Y-shaped constituent, we get a lower stiffness, the hydrogel, of almost only 4,000 Pascal. So you can see we can control the stiffness. What can we do with it? First of all, we can show it demonstrated visually. You can see this is the medium stiffness. When we go to the higher stiffness, a shrunken hydrogel is formed that shows a decrease in the volume by about 20%. But we can use this property of control stiffness to introduce some functions. For example, self-healing function. We can take the hydrogel of medium stiffness, cut it into two pieces, make it physically together to combine it physically, and then to apply the strand that makes it stiff. You can see that when we apply the trigger to make it stiff, we make the hydrogel smaller, but now, after a certain time interval of 3 hours, we see a self-healed hydrogel. The two pieces got together and got healed. On the other hand, if we don't apply this trigger E1, we remain with a medium stiff hydrogel, and when we shake it, you see a non-healed hydrogel. We can induce drug release from the medium stiff hydrogel upon going to the lower stiffness hydrogel that reveals very large pores. For example, if we load it with quantum dots, when we apply the trigger E2 that converts the hydrogel into the lower stiffness hydrogel, the quantum dots are released. And similarly, if we load the hydrogel with the drug like doxorubicin, we have the release of doxorubicin to the environment. Okay, so what did we learn until now from what I discussed? We learned that we can generate constitutional dynamic network, we can control by adaptivity their contents, and we can find certain application for such network. But we can go one step further and to move into networks that do not operate under thermodynamic equilibrated conditions, but under out of equilibrium dissipative conditions. And I want to show you a simple example of such systems. Let's say that we have a reaction module that is composed of two components, L1 and T1, and two scaffolds, F1 and F2, that are modified with two fluorophores. This is the rest condition of my network, If we apply a trigger on this reaction module, and we include in the reaction module also a nicking enzyme, the trigger L1 will displace this duplex to generate a duplex L1 prime L1 and will release the strand T1. T1 is engineered in such a way that it will hybridize with the scaffold F1 and F2 to form an intermediary product that has fret quenching efficiencies in the intermediate photoproducts. But the system is engineered in such a way that the duplex that we generate here is sneaked by the nicking enzyme into two fragments to release L1. If L1 is released, it displaces T1 from the intermediary complex and regenerates the original reaction module. So what we can see here is a system that exists in a rest condition. When we apply the trigger L1, we generate a fluorescent property of the intermediate properties, but the nicking enzyme takes care and regulates the system that autonomously the system goes back from its intermediate states. We have a temporal transient recovery of the original state. And this is a simple network that demonstrates dissipative transient properties. Certainly, these temporal transient properties are controlled by the concentration of the nicking enzymes, by the concentration of the trigger, and all this was verified experimentally. Now, what can we do with such systems? We can demonstrate that we can, for example, make transient system of enhanced complexity, for example, that demonstrate gated properties. If we take the reaction module that I just described and we introduce into it two fluorophore modules that are affected by the T1 that is being released. This is the reaction module. When we subject it to the trigger L1 prime, we separate this duplex. T1 binds to these two reaction modules. It releases the quencher units Q1 and Q2, and the two components become fluorescent. You can see here the transient fluorescence of the two components because the nicking enzyme is recovering the original reaction module. But let's say that we add to this reaction module, we add an inhibitor, I1, that binds to this scaffold. Under this condition, we get now a reaction module where this is blocked and only the intermediate transient operation of this reaction proceeds. So you see that we have a gated dissipative system of one component where the other does not show any transient activity. Similarly, we can use a second inhibitor that blocks this component, and then only this intermediate transient component is formed, and it generates this fluorescent. The other one is basically blocked. Now, what do we have here? We have here a dynamic network that is triggered by an auxiliary trigger. In this case, it was a strand, a fuel strand. And we have a regulator that controls the dynamic transient properties of the system. We can have different regulators for controlling the system. For example, we can have DNA Zymous controllers. Let's say that we take this reaction module, and in this reaction module, here that we see here, where is it? Here. In this reaction, we have a duplex and these two scaffolds. We have lead ions as co-component in the system. In the presence of the trigger, we separate this duplex, and the separation of this duplex generates this new duplex that is the hairpin with T, and the release strand B brings together the two scaffolds to generate an intermediate properties where the fluorophore Psi5 is quenched by BHQ2. But when we generate this duplex is basically a lead-dependent DNA zyme. The DNA zyme cleaves this strand T and regenerates the strand, the hairpin loop A, that displaces B and recovers the original state. So under these conditions, we have the formation of the intermediary state B, where the fluorescence is quenched, and we have the recovery that is induced by the DNA zyme. Again, we can control this dissipative, temporal, transient system in a gated way. We can now add two components, C and E, and the quencher unit as the reaction module. In the absence of any inhibitor, the two reaction intermediates, BCD and BED, are formed, and we get the transient operation of the true intermediates. But if we block selectively or one of the components or the second component with I1 or I2, we can dictate the gated operation of one temporal system or the second temporal system. Again, let me show that we have several such triggers to control the dissipative behavior of the system. And now I introduce the well-known that we addressed already, this azobenzene regulator unit. Azobenzene can be converted by light from trans to cis, and the cis azobenzene can undergo a thermal isomerization, back isomerization from the cis to the trans state. So if we take a Y-shaped DNA, where we have here a fluorophore and quencher as marker, in the trans configuration, this is the reaction module. If we shine light on it, the trans states is converted to the cis states, and we have separation of this duplex structure. The released G strand separates the Y strand, and once it is separated, you see that we can follow this separation by the fluorescence of Psi5. But the thermal back isomerization of the cis to the trans restores this Y-shaped structure through stabilization of this duplex structure. So again, you can see that you can have the fluorescent structure that is dissipatively recovered to the original state. And during this dissipative process, we can shine light from time to time and oscillate the system against to the separated, recovered state. Now, what can we do, again, with these dissipative structures of DNA And I'm going to show you a few possible applications. One is, for example, to release a load. Here we can see, for example, a DNA aptamer. This is the cocaine aptamer that includes here the cocaine as a load. If we apply a fuel strand on this cocaine aptamer complex, we open the hairpin structure of the cocaine aptamer complex, we release the load. The cocaine is released. But if we engineer in this strand a nicking site, and we have in the mixture a nicking enzyme, this strand is being nicked. After nicking, the aptamer strand recovers its original structure and rebinds the cocaine to the structure. So we have the temporal release of a load and reload of the aptamer with the cocaine load. We can have more complex dissipative systems, and this is an example that was developed, a nice example that was developed by Hanadi Sleiman in Canada. And what she demonstrated is that she can control material properties of DNA-based materials. And she can control it by pH that is induced using spiropyrin as a modulator. Spiropyrin undergoes a thermal ring opening to the protonated merocyanine isomer. This process involves the uptake from the environment of protons to make the protonated merocyanine. If we shine light on the protonated merocyanine, we get back in a very efficient and rapid way this spiropyrin, and this process releases now protons. So this is a regulator that releases protons fast and takes up the protons in a slow fashion. She used it to control the aggregation properties of polyadenine strands of DNA. At acidic pH, the polyadenine strands are dimerized to protonated adenine. But if this protonated adenine is subjected to the spiropyrin that undergoes thermal opening to the merocyanine, the acidity of the medium goes up. In other words, it becomes more basic. Protons are uptaken by the regulator. The polyadenine that was protonated got separated. But if we have in the medium cyanuric acid, non-protonated adenine and cyanuric acid forms a supramolecular complex that assembles into polyadenine cyanuric acid fibers. You can see here the fibers. Shining light on the fibers will, in the presence of our regulator, will transform the protonated merocyanine to the spiropyrin. Proton will be released, the fibers will be dissociated, and will reform the polyadenine structure. Now, the Protonated polyadenine shows a certain circular decreasing spectrum. The fiber have a different one, so we can follow the dynamics of dissociation of the fibers into the polyadenine and their dissipative reformation using circular decrease. You can see here nicely that when we shine light on the system, we form the polyadenine, protonated polyadenine, and it dissipatively forms the fiber, and we can repeat the process again and again by light and dark reactions. Here we can see a further example of application of such dissipative system, and it is on the aggregation of nanoparticles. We take a duplex L1T1 in the presence of two types of AU nanoparticles that are stabilized by nucleic acid 1 and 2. We have in the system the nicking enzyme. Under these conditions, we have a rest module system that includes individual particles. If we apply the trigger L1 prime, we displace this duplex to yield L1 prime L1, and we release the strand T. T bridges the nucleic acid functionalized gold nanoparticles and induces aggregation of the particles. Aggregation of the particles is accompanied by spectral changes as a result of the interparticle coupled plasmon excitation process. But we engineer the strand L1 prime in such a way that it is cleaved by the nicking enzyme. The nicking enzyme cleaves this strand, and the released L1 displaces the bridging units T1 to restore the original system. So what you can see here is you have a transient temporal reaction module where when you trigger the system with L1 prime in the presence of the nicking enzyme, you get a transient formation of an aggregate that is being dissociated because of the transient behavior of the system. This is exactly what happens. You can see here the spectral changes that are occurring as a result of aggregation, deaggregation. This is the spectral plasmon changes that show you aggregation, deaggregation. You can see at time intervals how the aggregation process proceeds by TEM imaging of the system. The aggregate is being autonomously deaggregated to the separated nanoparticle. Naturally, this dissipative aggregation, deaggregation process is controlled by the concentration of the enzyme and the concentration of the trigger. The last application that I'm going to describe the prior to the last application. It will be, again, the generation of a transient biocatalytic cascade, where, again, the confined medium controls the dissipative biocatalytic cascade. We have a reaction module of two strands, L1T1, and two strands modified with glucose oxidase and horseradish peroxidase. In the presence of the trigger L1, we separate this complex and we induce the formation of this intermediate structure where we generate an intermediate structure with a confined proximity of glucose oxidase and horseradish peroxidase that shows effective catalytic cascaded activities. The nicking enzyme separates this duplex, separates the intermediate complex, and restores the biocatalytic cascades to the original state. Again, you can see here that the dissipative biocatalytic cascade of the two enzymes is controlled by the scaffold of the DNA. The last example of application, I think, is a very promising, and it is an unpublished result yet. It is now being considered for publication, but I think that it is a very powerful demonstration how such dissipative systems can control transcription of messenger RNA. Now, in nature, transient gene transient transcription of genes controls many diseases and many processes in gene expression. So it is worthwhile to try and to develop a system that mimics such native systems. Now, how did we design it? Our system includes two transcription scaffolds that are shown here. This is one scaffold, and this is the second scaffold. We have in the system the RNA polymerase that basically transcribes the DNA templates into RNA products. The upper template, when it is transcribed, it generates the malachite green RNA aptamer that becomes fluorescent. Malachite green becomes fluorescent upon binding to its aptamer. The transcription of the lower template generates the RNA polymerase aptamer. The aptamer generated by this transcription scaffold inhibits RNA polymerase, so we have basically a regulator of the RNA polymerase. So in the rest configuration of the system, we have the polymerase inhibited by the aptamer. When we add the trigger L that eliminates this aptamer, RNA polymerase is activated and it induces the transcription of the upper template and the lower template. The upper template generates the RNA aptamer, but the lower template generates the RNA polymerase aptamer that inhibits the polymerase, and basically what it does, it acts as a suicider element that converts the upper transcription into a dissipative process. So you can see here that the rate of formation of the malachite green aptamer is controlled by the transcription and suicide generation of the RNA polymerase aptamer. The more we introduce, the more we enhance the suicidal effect, the lower is the transcription. We did the same story for Another aptamer, this is the so-called broccoli aptamer, and we were able to show that we can control in a suicide manner the second formation of the aptamer, and we were able to design a system that basically is a cascaded system that shows you that one transcription process of an aptamer can control a suicide transition on the second one, and we have a cascaded formation of two aptamers. I realized that my time becomes shorter and shorter, and therefore I want to move quickly to the last part that will be a much shorter part of my presentation. And this is only because I thought that this is something that you are more familiar to it. And these are DNA machines and structures that demonstrate dynamic behavior. Now, as I said at the beginning, we were very active in the past in designing DNA machines, we and others. For example, we were able, at the molecular level, we were able to design tweezers that by pH were converted from closed tweezers to open tweezers using the self-assembly of the arms into I-motif structures and their separation at neutral condition back to the closed tweezer. We were able to show a bipedal walking mechanism on a DNA scaffold using the reconfiguration properties of DNA. For example, we had 4 foothold where a bipedal walker was moving, and we moved this bipedal walker by metal ion complexes and by I-motif triggers. And I introduce here another example at the molecular level of a machine where the azobenzene, the photochemical azobenzene photoisomerization, is used to use a rotaxane DNA on an axle of DNA using light. This is a nice example demonstrated by Famulok, where he generated a rotaxane made of DNA that was bound to an axle and blocked by bulky units of DNA made of DNA. As an auxiliary unit, he used the azobenzene modified strand. When he photoisomerized it to the trans state, he generated a strand that displaced this ring to another position on the axle because of the strong interactions between the trans-azobenzene and the axle strand that was displaced by this strand. When he photoisomerized the trans back to the cis, he separated this strand and the ring moved to its original state. But I think that the nice machines and the nicer, I would say, perspectives that exist in the field use origami objects as a potential element to generate such mechanical machines of DNA. All of you know that we can, this is a revolutionary technology introduced by Rotemund, where he developed a method that can generate any structure of DNA using a computerized product that uses staple DNA units that staples today a long chain viral DNA. Now, these structures of origami units were used to dynamically demonstrate the possibility to place together objects of at the nanoscale. So the machines that were previously operating at the supramolecular level, what I have shown on one quick slide, all these technologies can be translated to objects at the nanoscale. For example, this is a very nice study by Sugiyama from Japan, where he took frames of origami tiles, and these origami tiles were modified with, at one edge of the tiles, with azobenzene DNA tethers. When he photoisomerized these tethers to the trans state, these units got hybridized and he got dimers of the two tiles. When he isomerized the azobenzene, the trans state to the cis state, he reversibly separated these dimers to monomers. You can see here nicely the dimer structure and the monomer structures of the tile induced by light and the mechanical transitions of DNA objects. We did a related study where we took origami tiles and we were able to dimerize and separate by, for example, pH. We designed dimers that were interlinked by bridges, where one of the bridges, L1, was rich in C bases. At acidic pHs, we generated I-motif structure and we separated the tiles. When we neutralized the system, we dimerized them. And we see here the dimers that were moving to monomers and back cycled into dimer structures. We were able to demonstrate that this method can not only be used to make dimers that are separated and reconstructed, but we can make isomerization of structure of the tiles. For example, from linear structure into bent structure. If we modify one of the tiles with two different types, that at one pH we have a linear structure, we get linear trimers origami structures. But when we move to we separate them at acidic pH to the I-motif, these tethers will bind to form triplex structure and form a band structure of our DNA objects. So you can see here that the reconfiguration properties of DNA can lead to dynamic movement of objects, and as expected, this reconfiguration of DNA was, for example, used in order to aggregate and deaggregate gold nanoparticles by pH using I-motif structures and dimer, or to aggregate and deaggregate gold nanoparticles by strands that with azobenzene form stable azobenzene cross-link bridged aggregates of nanoparticles, and under UV irradiation, we form the cis state that separates the particles, and one can reversibly switch on and off the structure of the nanoparticles. But what I want, and this will be my last topic that I'm going to, basically my last set of slides, that will terminate the lecture and will show you that we can even enhance the complexity of such DNA machine and objects to the macro nano scale. And I recall you about the possibility to generate structures of DNA that exhibits catalytic functions and catalytic functions that have DNA Zyme activity that cleave a strand of DNA. These are cofactor dependent DNA Zyme. Now what we did is we designed origami tiles where we can mechanically unlock a window in the tile of the origami and generate a hole in the origami tile. The aim of this was, and as we will see, to generate in the drilled hole, to generate a confined environment to perform a dictated catalytic function. Now, how operates this nano window on the origami tile? We have an origami tile. It includes a pattern of DNA that has here hinges made of origami, of DNA. The window is locked on the origami tile using two locks, L1 and L2. The window has two handles, and these two handles will help us to open the window. In the presence of the DNA Zyme, and in this case, it is a zinc-dependent DNA Zyme, the zinc-dependent DNA Zyme unlocks the lock, it cuts the lock, and two hairpins, H1 and H2, act as ropes. They bind to the handles attached to the window and open the window and link it like a rope to stators that are at the end of the origami tile to yield the open window. Now, this is the mechanical process that operates on the window, and those that didn't follow me will realize I have here a simple model that my coworkers designed. This is the origami tile. It has the window in the lock. When these are the two handles, and these are the ropes. When the window is unlocked by the enzyme, the ropes push it, and they put it on the stators, and to have an open window. Now here you can see nicely the closed tiles. When you open it, you can see here the hole, and you see here a bright spot. The bright spot is a high origami, a double origami thickness region that can be seen by the cross-section analysis that has the window lies on the on the origami tile. So we have a mechanism to open the tile. We did it not only with the zinc-dependent DNA Zyme, but also with the lead-dependent DNA Zyme. And you will immediately realize why we did it with the lead-dependent also. And we marked the tile of the origami that is opened by the lead with a hairpin structure that is this ball, so we can distinguish the tiles that are opened by zinc from the tiles opened by the lead. And again, you can see here the open tiles by lead. And now we can mix the two tiles of origami. And in the presence of zinc ions, we unlock only one type of the origamis. You can see here that we have the other one locked. With the lead ion, we open the second type of the origami, and we have the labeled unlock. And if we add zinc and ions to the system, we open the two types of the origami types. Now, what are we doing here in the origami by this opening of the window? We open a 15 nanometer hole in the origami tile. What we can do is we can now engineer from the beginning on opposite side of the open hole, we can add tethers that when we unlock the window, these tethers will be subunits of a DNA zyme, and in the confined hole, we will generate or a magnesium-dependent DNA zyme or a hemine G quadruplex-dependent DNA zyme, or we can generate the two enzyme activities. You can see here that we have or one active DNA zyme, or the second active DNA zyme, or both active DNA zyme. So what we did here is basically we used the mechanical properties of DNA to unlock a hole and make a reaction in a confined medium. But what is the disadvantage of this system? There is a disadvantage. The unlocking is irreversible. We have a lock that we break by the DNA zyme to make the window. The question is, can we make a lock that is reversible, that we can in a reversible way open and close the lock? You might be already after this lecture tired, but still sufficiently energetic to see, sure enough, based on what you say, we can design a lock that is made of azobenzene units, of trans-azobenzene units. In the presence of light, that we convert the azobenzene to cis, we unlock, and by this mechanism, we make a hole and in the system. And if we go back and to the cis state and unrobe the system, we can relock in a reversible way, make locking and unlocking of the DNA. And this is exactly what you see here. You see here the lock structure, the unlock, and by light, we were able to open and close the DNA structure and construct on the two sides a G quadruplex subunits that self-assembled upon opening into the G quadruplex and got separated upon closure of the window and get reversible, switchable, catalytic activity in this whole nanostructure in the DNA system. So I'm sorry for I took you such a long journey of dynamic DNA systems, but I hope that I was able to convince you that DNA is a fantastic and wonderful material to design dynamic structures of that can be used for different purposes. And what we have seen here, that we can emulate native dynamic network by making either constitutional dynamic networks or dissipative transient dynamic network. We have seen different applications of such systems, and I believe that there are many other possible applications of such dynamic network. The question is only where to go with such networks. And I feel that if we want really to emulate and to mimic natural processes, the next step is to try and to integrate all these networks into cell-like environments, or what is called a protocell. And today there are tremendous activities in the area of protocells, and I believe that the integration of these dynamic networks into protocells will really lead to some kinds of artificial life and artificial cells. In the second part of the talk, I think that I convince you that we made as scientists a significant and important advance in moving from molecular and supramolecular DNA machine into nanostructures that can be manipulated in a dynamic functions and functional, and finally into structures that are nano devices, and at the macro nanoscale can be considered as the smallest apparatus and devices for chemical reactions. I thank you for your attention, and I will be glad to answer any of your questions. Thank you.
Thank you, Professor Wilner. Very nice talk. Now we open for discussion. We will ask Ms. Professor Meng Jiaocheng to repeat the questions in English.
We have collected several questions, Professor Wilner. The first question from Professor Zhenggang Wang. He mentioned your work on ACS Nano in 2020, the transformation of single-strand DNA into single-strand DNA outside the pyramid can release the encapsulated drug like CPT or DOX. He asked, what is the mechanism? DOX should also have been intercalated within the double-strand DNA.
Well, I didn't mention it. This is not simple DOX that was in the DNA. If I recall properly, it was DOX that was covalently linked tethered to cellulose, and so it does not intercalate into DNA.
Hello, Itamar.
Hi, Zhenggang.
Hi, yeah.
Good to hear you.
Yeah, good to see you. I wish you can come to China again in the very near
future.
Sure,
I'm looking forward. I'm homesick.
Yeah, we all look forward to your visit to China again.
Thank you, Zhenggang.
Okay, so Professor Chen, you can move to the next questions.
没开麦克。
Sorry. Let's move to the next question from Chen Wang. Life is operated under out of equilibrium conditions. What's your opinion on the next step for the development DNA-based out of equilibrium systems? And how further can we go by these artificial dissipative DNA systems? and what's the specific functions and advantages as compared to thermodynamic controlled processes?
Okay, this is a very, very interesting and very important question. And I think that the systems out of equilibrium have even a much higher potential and perspectives for the development of systems like that have cell-like activities. In my opinion, to try and to make dissipate that, I mentioned one slide that is the transcription of messenger RNA. And we are not there yet, but we are very close to be there. And I think that coupling the transcription under dissipative conditions to the translation of proteins, this is the way to go. Because if you can make transient proteins and transient different proteins through gated mechanism, then you can really reach in the cell, you can reach cell-like functionalities. Now, I think that the in vitro the translation of the RNA in vitro test tube systems is relatively easy. But one has to find a way to integrate these dissipative systems into cell-like environment. And once you are doing this in the cell-like environments, you will be able to mimic life.
Then the next question from Dawei Zhang. Very interesting talk and fantastic work. I think a big challenge in system chemistry is the characterization of the system, especially in your talk, and DNA was used, which I guess make it more complicated. What kind of techniques have been used for characterization and how reliable are they? Thanks.
Okay, this is again an excellent question that I didn't have the time to dwell upon. But if you look at the paper carefully, and especially each of these papers is usually accompanied by 60 or 80 pages of supporting information. And this part is usually addresses exactly this question that I was asked now. Now, I emphasized from the beginning, the characterization is usually optical through the transduction by a DNA Zyme component in the equilibrated system or by fluorophores, quencher units in the dissipative system. This is usually the most important way to characterize the system, but each of the systems is usually accompanied by very tedious gel electrophoretic and quantitative gel electrophoretic experiments that can follow the contents of the constituents either in the dissipative process or the equilibrated system. Furthermore, and this is something that I am very often, I would say, after publishing the results, people praise this accomplishment. All our systems, either dissipative, but I would say most of them, dissipative or equilibrated, are accompanied by kinetic models. You have, in all these systems, you have, in a simple system, you have 25 to 30 rate equations, but in more complex systems, you can accumulate about 60 rate equations. And we simulate our results by computational, but we simulate the computational, the kinetic model by computational simulation. And we derive rate constants. Now, these rate constants that we derive guide us and tell us, okay, take now the CDN or the dissipative system at other conditions that you didn't explore and predict by the computational model how will look the result, the expected result. And when we do the result, usually, if everything works well, the predicted result is fully supported by supports the experimental results. In other words, we are able to predict the results and to confirm the results by the experiments. Now, this is not enough. And I tell always my coworkers that when they do such computational modeling, they have, out of the 60 rate constants, they have to evaluate at least 2 experimental rate constants from this jungle of rate constants. If they are able to confirm that the experimental rate constant coincides, overlaps with the computational, then you can rely with the computational results. So what I say is that usually we have here a combination of physical characterization of the system by optical and electrophoretic means, and then computational analysis of the results and validation of the computations by experiments.
Thank you. Then the next question from Professor Mingfeng Wang from the Chinese University of Hong Kong in Shenzhen. He asked in the part of DNA origami assembly, how to overcome the errors or defects in the assembly.
Well, this is again a very good question. Every study that, especially mechanical studies with DNA, with origami, will be accompanied by defects. You cannot overcome these defects. The only thing that you can hope is to optimize the system to reach a situation with minimal defects. But in all of the systems, we always have structures that I would say we cannot decide whether they are open window, closed window, whether they are dimers or monomers, whether they are trimers from some reason or dimers. So in the counting and the statistical analysis, we always take into account this problem of defects. I would say that on the average, in the most complex systems, we have at most 10 to 12% of defects in the structures, not more. So about 88% to 90% of the structures are really defectless structures.
Thank you. We look forward to have very nice systems like mismatch repair in life, in real DNA replication process. Actually, life has a toolbox to fix defects or errors.
No question.
Yeah.
A great challenge.
Yeah. And the next question is from Yang Xia. The question is quite specific about the DNA enzyme. After the DNA enzyme cuts the substrates, does the substrate fragments have to dissociate from the enzyme or still binds to the DNA enzyme?
Well, from our point of view, they have to separate. I mean, we try to design, and this is one of the important issues, the design of these systems and the evaluation of the relative stabilities of the systems. So I would say that we try to design the system in such a way that the fragmented DNA enzyme will be separated from the backbone of the DNA enzyme. But this is a matter of, first of all, temperature, ionic strength, but more important, the temperature and ionic strength you can optimize and you can regulate. But you cannot fight thermodynamics. And if the concentration of the fragmented products is high, you will always find yourself with an effect of rebinding to some extent of the fragments to the DNA enzyme. So at high concentration of the fragmented DNA enzyme, you will have inhibition of the DNA enzyme.
We still have many questions, but it's a long time for you. It's almost 2 hours. So the last question is about the application of DNA enzyme to detect small molecules like antibodies-based aptamer. What details do I need to pay attention to when designing DNA structures? Quite general.
Well, this is a very, very, I would say, I could talk about it 1 hour, yeah. 1 additional hour, how to design the DNA enzyme and the aptamer sequences in order to get the best sensing performance of the system. And even after giving a lecture on this of 1 hour, I will be probably mistaken, and I will have some errors that will be needed for further optimization. So I would say that in general, I would say that first of all, regarding aptamer ligand interaction. And here, this is something that is quite advanced these days. There are excellent softwares to predict the expected binding affinities between an aptamer and a protein or a small ligand. and what will affect. So a lot of money can be saved and ordering DNAs, and a lot of experimental efforts can be saved by making these simulations, using the softwares to predict. The DNA activities is more difficult, and it depends very much what DNA enzyme the person is using. I personally would suggest as the best DNA enzyme to apply for sensing applications, or the magnesium or the zinc dependent DNA enzyme. Recently, we apply the histidine dependent DNA enzyme, so I would recommend to select one of these 3 DNA enzymes.
Thank you. Thanks for your nice talk and answering these questions, and for great support of this journal. So I moved my time to Professor Feng Shi.
Thank you very much, and thank you, Professor Meng Jiaochen, to help to repeat the question. Now we will move to a one-to-one talk. And before that, I would introduce the next week. Next Friday, we will invite E.W. Mayer to give us a talk about,
Superpolymer. Bert Meyer from Netherlands.
Yeah, Bert Meyer.
Yeah.
From the Netherlands.
Bert Meyer. The Chinese topic is 功能超分的体系及材料. The English one is do you have it? Sorry.
Shall we have a short break before the one-to-one talk?
Functional supramolecular systems and materials.
Yeah, functional supramolecular systems and materials. Hope next week we can see all of the audience. Yeah, thank you very much.
Thank you, Professor Wilner. So, shall we take a break, like 10 minutes, and then we move to
problem. We move to another link.
Yeah.
Okay.
One-on-one talk.
Okay. In 10 minutes, we meet again.
Okay.
Thank you.
Bye. Thank you.
Bye.
Thank you.
Thank you.
Yeah.
Bye.
Bye.